The MIT Edgerton Center presents Protein Folding in Water, to be used with the MIT Edgerton Center protein set. Every protein is a folded chain. A protein's job requires a unique shape. Protein folding begins with the correct sequence of amino acids. Build the protein chain on page 11. Keep the black amino end to the left as you build. The eye of each amino group should be showing. Otherwise, the chain cannot bend. Turn all the amino ends so the eyes show. Turning the ends helps the model to fold. Check the rest of the chain. How will the protein fold in water? Hydrophobic side chains hide from water. Hydrophilic side chains are attracted to water. Gently fold the chain using this information. Hydrophilic side chains turn to face the water. Hydrophobic side chains turn towards each other on the inside of the protein. The hydrophobic amino acids are clustered together in the center. The hydrophilic amino acids are on the outside of the protein. This protein chain is folded using only one rule. Several rules are required for the unique shape of each protein. More resources at edgerton.mit.edu/DNA-proteins-sets.